So this is the lecture over DNA, and what we want to do is take a look at DNA, a little bit about the history of DNA, how it was discovered, and what we know about DNA. Now, something I will definitely want you guys to make sure you're aware of or comfortable with, we really don't know everything there is to know about DNA. We know it's the genetic material for life. That's what we assume at this point. That's the basic premise. We know of information called RNA, which we'll get into in later lectures, but there's still a lot of uncertainty about DNA. We have a great foundation, and the scientists and the folks who've worked on this for 100+ years have done a great job in discovering a lot of this. But there still is a world of opportunity, a world of new information that needs to be discovered as we look at DNA. So I want you to keep in mind, when we talk about genetics and DNA and biotechnology, this is our current knowledge. It's always possible that it will change as we discover new things. Okay, so let's take a little bit of a tour through the history of DNA and look at how scientists got to the point where we're at today. And then what I really want you guys to think about is, where do you think we could go with this? What's the future of genetics and DNA and research and things that we may want to do down the road? Okay, so when we look at DNA, we can jump back to 1928 and take a look at a scientist named Frederick Griffith. So in 1928, he was working with Streptococcus. Now, guys have ever gone into the doctor for a sore throat? Sometimes that's caused by Streptococcus, strep throat. And Griffith was working with two strains of Streptococcus, what he called the S strain, that is virulent. And virulent means lethal, deadly, nasty, nasty stuff. So let's make sure we're comfortable with some of the terminology here. Virulent, bad stuff. Can be, not always guaranteed, but can often be deadly. Non-virulent, not so bad. I mean, it's there, it's in the environment, but it's not usually going to kill you. So the R strain is non-virulent, the S strain is virulent. And what Griffith did is he worked with mice, which are often a test subject, often a organism used in research, especially in molecular research, and he helped sort out how this stuff works. Okay, so what Griffith did, he inoculated mice with the live S strain, the virulent strain, and that killed the mice. Okay, so you got dead mice there. He inoculated or injected the bacteria into the mice, the live strain R bacteria into mice, and the mice lived. He went, okay, so we know which one's deadly. We know which strain of Streptococcus is deadly. So he was working primarily with that. So what he did is he took the S strain and he killed the bacteria by heat treating it. Basically blasted it with heat to kill the bacteria. Injected that into mice, and the mice lived. So dead bacteria that is normally virulent when it was alive, injected into mice, mice did not die. But then he took the dead S strain bacteria, heat treated it, killed it, which in the previous experiment did not kill the mice, but he combined the S strain with the live R strain bacteria. So mixed it together in the same petri dish, the same culture medium, and then he took that mixture, injected it into the mice, and it killed the mice. So the question was, well, wait a minute, dead bacteria, S strain, doesn't kill mice, live R strain doesn't kill mice, but mixed together, this kills mice. So he had a big question. Hypothesis. What's going on here? All right, so here's what we're seeing. Live virulent strain, S strain, kills the mice. Live non-virulent strain, the R strain, doesn't kill the mice. When he heat treated it, the mice lived. When he mixed the heat treated deadly bacteria and he took the live non-virulent strains, mixed them together, the mice died. the lungs actually contained live pathogenic bacteria, the S strain. And he, again, came up with a lot of uncertainties. So what he discovered with his research was this idea or this principle called transformation. Virulence can be passed from one cell to another, especially in the bacterial world. So this deadliness, this toxicity, this ability to kill, virulence, can go from one bacterial cell to another. It can do it when they're alive, but in his case, it was doing it when they were dead. So Griffith, I don't want to say coined, but came up with this concept back in 1928. Now, springboarding off of that, using some of that information about, well, what is it that's moving from the dead bacteria to the live ones or being picked up by the live ones? What is this stuff? We move to Avery, MacLeod, and McCarty. So these scientists were working in 1944. So I want you guys to think about what's going on in the 1940s. What global phenomenon, global event is happening right now in 1944. So science was still continuing, going in all sorts of different directions. So this is where you bring history together with science. And in 1944, Avery, MacLeod, and McCarty actually identified DNA as a genetic material. So they're the first folks to really identify DNA as a genetic material. Now, most everybody has heard of Watson and Crick. Very important characters in this picture. But these guys are almost a decade before Watson and Crick. So they identify DNA as a genetic material and are really helping set the stage for the next level of genetic research. Their main focus was looking at bacteria, bacterial DNA, and that is what moves from one bacteria to another through transformation. It moves specifically in these little loops here called plasmids. We'll get into this a little bit more. As you guys move to Bio 112, you'll definitely get into this a lot more. That's where we get to spend a lot of time on bacteria and explore what's going on with that. Okay, so we've gone from the 20s to the 40s. Again, what happened in 1944 will connect into more of the biotechnology unit, or I should say what happened in the later 1940s, 45, 46, etc. And now we want to jump into 1952. Hershey and Chase. These scientists discovered these things called bacteriophages. Bacteriophages are viruses that specifically infect bacteria. They go after bacteria. They will not go after a human. They do not recognize our cells. So if you take a look at the picture here, and some of these bacteriophages are just awesome looking. It's this incredible looking little structure, they look like little robots or space aliens. That's the actual structure. So this thing we're looking at here, this is a bacteriophage right here. All the little stringy things up here, that is its genetic material. And the reason these guys are specific to bacteria are these little pointers on the tips of their legs here. These little pointers are proteins, protein structures. So I want you to go back to chemistry, and we talked about protein, and the shape of the protein determines the function. Well, those little pointers are what allow that bacteria to actually that virus. Those pointers allow that virus to identify the bacteria. That pointer, if it hits a human cell, doesn't identify it, doesn't recognize it. It specifically identifies and recognizes the certain bacteria. And it's not all bacteria, it's specific types or strains of bacteria that these certain viruses will infect. So Hershey and Chase played a role in helping discover these things we call bacteriophages. So the way this works is take a virus like Ebola or SARS or West Nile virus or Zika, and that virus attaches to the cell. It uses those little pointers, those little hooks to attach to the cell. It then injects and enters its genetic material into the host. That genetic material inside the host gets copied. We call it synthesis of the viral genome. So the virus takes over the host and uses the host to make more copies, and depending upon the type of virus it is and the life cycle, it may assemble new viruses and they then rupture and explode out of the cell and go after new cells, continuing to attack, or sometimes they simply, the viral DNA gets blended into the host DNA. So this happens with viruses when they're going after bacteria, when they're going after people, etc. So if you guys get the flu, viral infection, that virus is destroying your cells, which is why the flu sets in and you feel horrible, but then your immune system hopefully kicks in and gets rid of the virus. If anybody has had chickenpox, the viral DNA to cause the expression of chickenpox is in your body and it will not leave. It is blended into your genetics and it will be with you the rest of your life. And later in life we worry about shingles because that virus comes back as shingles, but it's with you all the time. And what this is revolving around is the idea that the DNA carried by the virus is now inside your body. It's the genetic material that is directing what's happening at the cellular level. So the viral DNA sometimes takes over and hijacks our cells and does stuff we don't like. Okay? So let's take a look at DNA itself. DNA is known as nucleic acid. Okay? So all living things that we know is made up of, its genetic material is made up of DNA. RNA can be found in viruses, but viruses are not classified currently as living organisms. But all life, from bacteria to plants to birds to fungi to people, DNA is the basic code. And DNA is composed of nucleotides. Now the picture here shows the basic nucleotide. It has 3 components to it. So fill in the blanks here. 3 blanks, 3 components to the nucleotide. The first component is this deoxyribose sugar. So right here, call it the 5-carbon sugar. That's what deoxyribose represents. Each of these points represents a carbon. 5-carbon sugar, you could say it is a sugar shack. I always thought it looked like a little house. The house has a phosphate group attached to it. That's the phosphate up here. So phosphate, like a chimney. So here's your sugar shack or your house with a chimney. So the second component is the phosphate group. The third component is a nitrogenous base. This little base here that sticks off, kind of like a porch. So you have a house, a chimney, and a porch. So 5-carbon sugar, phosphate group, nitrogenous base. Now, with all living things, there are 4 options for your nitrogenous bases, 4 options for your porches. The option 1 is adenine, that would be right here. Option 2 is thymine. Option 3 is cytosine. And option 4 is guanine. Okay? So those are the 4 options for the porches. And Friedrich Miescher actually was one of the earliest folks to discover DNA back in 1869. So he kind of sorted out what this was, but really didn't understand completely how it works, what it can do, the differences between species. That has taken hundreds of years, or 140 plus years worth of research to get us to where we're at today. Okay, so when we're looking at DNA structure, here's it again in a slightly different version. Okay, so our slightly different version still shows sugar shack, phosphate group, nitrogenous base for a porch. In DNA, what we're looking at is that 5-carbon sugar, right there. And the porch options or the nitrogenous base options include adenine, thymine, cytosine, guanine, ATCG. When we look at RNA, a little bit different. We're going to kick out the thymine option, replace it with uracil. And we'll talk more about RNA at a later point.